Hey everybody, it's 8 o'clock, so it is live with Fiveable. You are here for the AP Biology stream on cellular respiration, so thanks for logging in. Okay, we'll just get started, and everyone else will just kind of have to catch up with where we're at, and also there's obviously, you can watch the streams over again and get access to the PowerPoints. Tonight, we're going to do a quick photosynthesis review. I just have one slide just to kind of summarize, and then 4 W's of cell respiration. So we'll be looking at what is it, who does it, where does it happen, and why does it happen. And then really just getting into what's chemical energy and how does it become cellular energy. Okay, so review for photosynthesis. Photosynthesis is our conversion. We have light energy. So in this picture here, it's showing light energy being made into sugar at the very bottom, right? Obviously, there's a lot that happens in between that, but big picture is conversion of light energy into chemical energy, so sugar. And chemical energy is something that we're talking about bonds within molecules, right? And sugar is just the example here of what plants and other autotrophs can do using their chloroplast in photosynthesis. Sorry, my screen's a little messed up. Okay, where does it happen? Okay, so where photosynthesis happens, if you look at this plant cell here, can someone type in what's the name of the organelle, those little green things inside this plant cell? So again, we're just doing a quick review. Awesome. All 3 of you, great. So yes, the chloroplast is where it's happening. It's obviously happening in either a plant cell or it can also happen in some protists, which we'll get to later, but things like algae and kelp. And there are also some photosynthetic bacteria. So it's not just plants that can do it. So a lot of people, a lot of students will zoom in only talking about plants. But if you have a free response question of sorts that's asking about what kind of organism can do this, anything that could be labeled as an autotroph or an autotrophic organism. So again, protists, photosynthetic bacteria, and you'll get more into protists and things like that later on. I usually teach that kind of stuff second semester. So when I say protist, if you're like, what is she talking about? You'll learn more about that later, okay? I like this picture here to kind of help us zoom in on the actual plant cell and chloroplast. So if I were to zoom in on one chloroplast, it's like this little Pac-Man shaped thing right here, and then it has these green stacks of, they kind of look like green pancakes. So that is zoomed in on the chloroplast, and inside the chloroplast, we have these stacks here. And so it's important to be aware of where the parts of photosynthesis are happening. Okay, so obviously it's happening in a in an autotrophic cell, right? Which is happening in photosynthesis is happening in the chloroplast. But the 2, I guess I would say, structures within a chloroplast that you should be aware of are the terms stroma and thylakoid. Okay, so thylakoid, I have the arrow here pointing to those green stacks of pancakes. That's a very membranous structure. It's got lots of different folds. And we'll talk about that in a second, why the folds are important. And then the other section is the stroma, which is basically the cytoplasm of a chloroplast. So let's just call it a plant. Let's say you have a plant cell. Okay, inside each cell of a plant, you've got all sorts of little chloroplast organelles. Okay, within that organelle in the chloroplast, you have cytoplasm, right? So that's kind of filling the chloroplast, like a cytoplasm fills a cell or any cell. So the stroma is the cytoplasm of your chloroplast, and then the thylakoids are those stacks. And so if we think about where the 2 reactions happen, your light reactions, that first reaction where light energy is captured, that happens in the thylakoid, and then the second citric or the Calvin cycle is happening in the stroma. So just be aware of where things are happening, which sections are going on here. So again, for those of you who are just joining, hi, welcome. I know this is a big topic, cellular respiration, but I just wanted to start with a review of photosynthesis, just really briefly. And a big reason is that our reactants from one process are the products of the other. So for example, here I've got my drawn equation for photosynthesis. So my reactants are carbon dioxide and water, and your products are sugar and oxygen. this is something that you maybe even learn in 5th grade, right? So why do we need to go over it in AP biology? You should be able to defend why it's important and why, like, what's going on with matter and energy recycling for an organism that does both photosynthesis and cellular respiration, right? So if I'm a plant cell and I have chloroplasts and mitochondria, why is it so important that they're able to recycle energy and matter, right? So if a plant is releasing oxygen, where can that oxygen go right away, right? That oxygen can be used right away as a reactant for cellular respiration. Okay, so just make sure you're aware of your reactants and your products for both of these things. Okay, all right. Also, for those of you who maybe missed the photosynthesis stream, we are hoping to do another one that's just focused on like question and answer. So I did the photosynthesis stream last week, and I would love to host another one where you can just blast out questions about what's going on with photosynthesis. So keep your eye out for that. If you could give me an emoji of like a thumbs up or a high five or something, that is something that you would want, go ahead and do that. Like Sophia saying yes, Sandra saying yes, or you can just give me an emoji. Good. Okay. Yay. Yeah, I like the idea of doing that, of just kind of hosting an open-ended. I can prepare a couple free response type questions. So if people get stuck and they don't have any questions, maybe going over a free response question will make you have questions. Okay, so that's another really good way to study, is know ahead of time what kind of questions you have about photosynthesis. So again, we will, it looks like a lot of you are interested in that. So we'll try to get that scheduled sometime in the next couple weeks. Alrighty, so, oh shoot, I have a typo here. It says the 4 W's of photosynthesis. That's bad. Hold on, let me fix it, because that'll bother me if it's not fixed. All right, much better. Sorry about that. Okay, sorry. So 4 W's of cellular respiration. I just kind of came up, I've never actually lectured that way before. I did not know. Anyway, I've never lectured about that before, like calling it the 4 W's, but as I was planning tonight's session, I thought, well, these are kind of the 4 big things. If you're able to walk away and tell me what is cellular respiration, who does it in terms of living organisms, where is it happening, and why does it happen, then I feel like you're on at least a good start to understanding it. Another important thing that I've mentioned in some of my streams is that I would make sure, by the way, great job taking the time to be in a stream and to help yourself expand on AP biology knowledge and help you prepare for the test. But also, in one stream about cellular respiration, don't expect to be an expert right after this, okay? So if you leave a stream with more questions than you had before, that just means you're learning something, okay? I have a lot of students who get frustrated because they're confused and they feel like they don't know enough, but that's part of the learning process, okay? If everything just came easily, you wouldn't be learning anything, right? It would just be like, oh, okay, yeah, that makes sense, moving on. So if you are in a rut, or if I call it the pit, or if my students feel like they've fallen into a pit and they can't get out and it's confusing and their mind is just, I know you're all in a lot of other AP classes, let yourself be in the pit for a little bit, okay? Like, be okay with the discomfort of being confused. And so be realistic about, setting aside time to work on it. So if after tonight you're still wanting more practice with cellular respiration, go do that, right? Pick a part of cellular respiration and go back and read over your notes, or ask your teacher, right? Or go to Khan Academy. There's a lot of things out there that can help you, but instead of becoming a passive learner where you're just sitting back and listening, which is a good thing, that's part of, being a learner is listening, but also making yourself think and being okay with being confused. Okay, so anyway, that's my rant about learning. Okay, so what is it, right? What is cellular respiration? I always like to start with just the equation, right? So if you're able to talk out the equation and tell me what it is, right? It's the process of converting. Now we have chemical, right? An actual chemical substance, chemical energy. We have glucose, and we're harvesting that to make actual usable energy, right? So some people would say it's called like it's stored energy to usable energy. But if you think about like if I'm having low blood sugar, right? If anyone's interested in anatomy and medicine and all that, this is a good example. So if I feel like I have low blood sugar and I'm feeling just tired and lethargic, and I choose to snack on an apple, right? There's sugar, natural sugars in an apple, right? That's energy, but my body just can't, once I chew it and digest it, it's not just immediate energy, right? Your body has to do a process, cellular respiration, in order to make ATP. And then ATP is the usable energy form that allows your body to do cellular work. Okay, so, and then oxygen, obviously, if you're ever confused about knowing where does oxygen play a role in cell resp or photosynthesis, just think about you as a human. If someone's taking like your respiration rate, right? It's that breathing respiration. So think, okay, well, I know I have a respiration rate. I breathe in oxygen. So that has to be a reactant of cellular respiration. And then similarly, you breathe out carbon dioxide. Okay, I think that's something that we all learn at a very early age, that we breathe in oxygen and we breathe out CO2. And then also, a parallel to that is that you think of plants and how they breathe out oxygen and take in carbon dioxide. So we're perfectly matched in terms of being able to use each other, living organisms that are producing a bunch of CO2, and then photosynthetic organisms that need CO2 to do what they need to do. So it's a very beautiful relationship that's not by mistake. It's very, very cool. Okay, so big picture, we're going from glucose to ATP. Okay, who does it? Who can do cellular respiration? Technically, all living things are able to take something like glucose or chemical bonds, break them up to make ATP. Okay, if you are an organism that does not have a mitochondria, you're not technically going through the entire process of cellular respiration like we'll learn tonight. They are stuck in a way of just doing glycolysis. But if you're a small organism like a bacterium, making 2 ATP for every molecule broken is actually not that bad, right? So evolutionary speaking, if you're a very, if you're an older organism, right? So you're more, I'm totally blanking on the word. Sorry, I didn't eat enough. I was talking about an apple earlier because I think I do have low blood sugar right now. Anyway, evolutionary speaking, they're more ancestral, so older, I guess, in terms of like the origin of life. You're gonna at least be able to do glycolysis, and that's that first step of cellular respiration. So that's who does it. Every living thing does something to make ATP. Okay, where does it happen? So if you can do all of cellular respiration, it is happening. Oh no, I didn't mean to do that. It is happening in the mitochondria, which is like this little squiggly red thing right here. Okay, so it's happening most of the time in the mitochondria. That's why when you learn about organelles, we talked about the mighty mitochondria. I'm sure your teacher had some way to help you remember the mitochondria, but most people do mighty mitochondria. I'm typing that in. Energy source equals ATP. So those are kind of like the big buzzwords for the mitochondria. And then why? I already kind of alluded to this. Why? It's to make ATP. And ATP, I might even do like a mini session just on ATP because I could go on about it for about a half an hour about how it's used and why it's used. And it's something that most people recognize it as an energy molecule, but they don't really understand the actual how that happens. All right, so 3 stages of cellular respiration. So the first stage is glycolysis, sugar, lysis. So lysis means cut, okay? So that is happening in the cytoplasm. Okay, so here's another picture of our mitochondria, but outside of the mitochondria is your cytoplasm. So glycolysis happens there. The second stage is called the Krebs cycle or the citric acid cycle. Your teacher might use one or the other, so just be aware of that. If someone starts talking about the citric acid cycle, it's the same thing as the Krebs cycle. And that's going to be happening in the mitochondria, in that inside mitochondrial matrix. Okay? The electron transport chain is we know from photosynthesis that the electron transport chain that happens in the very beginning. So when we have the light reaction in photosynthesis, where you have those two photosystems that are going to be taking in the light energy, and they excite an electron, and then the electrons fall down through this membrane. It has to happen across a membrane, right? So in photosynthesis, it's happening across the thylakoid membrane. Here, it's happening in this inner mitochondrial membrane. So you can kind of see here, I'm pointing to this inner membrane. That's the membrane that the ETC, or electron transport chain, is happening. So it's inside the inner membrane there. And I would encourage you to draw pictures of stuff as you're learning it. Drawing pictures is a really good way to help you make sense of all this stuff, all this information. Yes. Okay. I see that Joseph has asked a question. So I should have told everyone this in the beginning. If you do have a question, you can go to the thing at the bottom that says ask a question, and then other people can be looking at what questions have been asked, and they can vote for a question. So for example, Joseph's question says the products are H2O and ATP. You could vote and be like, yeah, I want to go over that too. So start answering. I'll answer his question. The products are going to be water and carbon dioxide and ATP. So carbon dioxide is also made in cellular respiration. Okay, so again, if you have a question, type it in. All right. Oh, the Krebs cycle. And usually I'll wait till the end for the questions, but because I'm talking to you about it now, I might as well answer the two that are there. So Soleil, or I can't remember, is it Soleil or Soleil? Just let me know. Where does the Krebs cycle happen? It's happening in the mitochondria. So like the very inside of the mitochondria. Thank you, Soleil. Okay. Inside the mitochondria. Technically the mitochondrial matrix. So the most inner portion of the mitochondria is where the Krebs cycle is happening. Okay, really good questions. All right. So here's just a big picture of cellular respiration. So don't freak out. I'm going to talk about each individual part separately. Okay, so we're just getting an overview right now, but I like this picture because it helps us reinforce where things are happening. So here in this bluish teal color, it's showing that glycolysis is happening in the cytosol, or the cytoplasm. So we're outside of the mitochondria. That's where glycolysis happens. The reason we focus on where it's happening so much is because then if we learn about an organism that does not have a mitochondria, for example, if there was a free response that said, a grad student found this organism that was able to produce, it was producing carbon dioxide. They were able to measure carbon dioxide and, defend whether or not it had a mitochondria, right? And so you'd have to really know, okay, well, where does carbon dioxide get released in the process of cellular respiration? And if it's not in the cytoplasm, if it's in the mitochondria, then that would mean that organism has a mitochondria. So I just came up with that off the top of my head. It's not a great pre-response question, but if you get the idea of knowing where things are happening, you'll be able to use that as evidence. That's a huge thing with the College Board, that you're able to not just, they don't want you to be able to, throw out facts. They want you to be able to reason through ideas. So if they tell you, hey, this is a statement someone's making about some process, are you able to, as a student, as an AP biology student, are you able to take information that you know and use it as evidence to defend or, shut down an idea? Okay, so kind of think of that too. I know it's a lot and you want to just memorize everything, and that's how I was as a student. I just wanted to memorize it and regurgitate it, but they're asking a lot more of you. They've changed the exam, and you need to go deeper, and you need to be able to reason through why things are happening. Okay, so anyway, so glycolysis is step one. This peach box that's called pyruvate oxidation, it's more of an intermediate step. It's not, some people might say, oh, I like to think of it as 4 steps. We've got glycolysis, pyruvate oxidation, citric acid cycle, and then the electron transport chain. But really the pyruvate oxidation is literally one step. it's so minor, it's so small that it's not, compared to all the other things, it's an intermediate step. It's really just changing pyruvate so that it's able to enter the citric acid cycle or Krebs cycle. So just FYI, if you see pyruvate oxidation, you don't worry too much. It's just another good example of how enzymes are used to make these make a process like this happen. Because after all, we just learned about enzymes, right? And so enzymes help catalyze reactions. And so it's just another helpful example of an enzyme being used. Okay, so we're inside the mitochondrial matrix, which is the inner portion of our mitochondria. The citric acid cycle happens. Big picture here is that electrons are then carried to the last step, the electron transport chain. So it's called electron transport. So there better be some electrons that were brought there from the previous cycle. And here's where the big, if you notice the ATP here, at the far right is a bigger star. That's where the bulk of your ATP is made. A little bit of ATP is made in glycolysis. A tiny bit of ATP is made in the citric acid cycle. But really the biggest thing is the electron transport chain in chemiosmosis. That's where ATP is made there. Okay, so we're now going to break down each of these steps and try to focus on what's being oxidized or reduced, and then what's going on with the electrons. So try your best to follow the electrons. Okay, that's a big thing, is follow the electrons. I've put in a blank copy of this for you if you'd like. Just because sometimes if you don't want to draw your own diagram, it's nice to have a blank diagram to work with. Again, this is not going to help you memorize any steps or things like that, but it'll help you conceptualize where things are happening. Okay. So real quick, concept check number one. Number one, the primary purpose of cellular respiration in living organisms is to A, remove excess carbon dioxide, B, create water by combining oxygen and hydrogen, C, produce biologically useful energy in the form of ATP, D, produce molecules that can be used in the electron transport system, or E, create larger complex molecules such as carbohydrates, proteins, and lipids. Good. Yeah, C. There's a couple distractors in here. The answer is C. Good. So produced biologically useful energy. That is the reason for cellular respiration. A couple distractors that were in here, I would say, is B, that create water by combining oxygen and hydrogen ions. That does happen in cellular respiration. That's the very last thing that happens. Water is made because oxygen and hydrogen are combined, but that's not the purpose of cell resp. Same thing for D. Molecules that are used in the ETC, that does happen in cell resp, but that's not the big picture, right? The Krebs cycle is what produces molecules that can be used. So just watch out for those things and don't just choose the first thing that looks right. Make sure the right answer is answering the right question. Okay, so glycolysis, it is a much more complicated series of steps. But this is kind of the Cliff's Notes of it. Many years ago, the AP biology class, you probably would have had to have all the steps memorized, and then you would just had to regurgitate it. Instead, it's kind of out of the scope of the class now to memorize all those steps. It's more of, okay, what's the big picture? Where is it happening? What's the big picture? So I have a picture here of bread being cut because, there's sugar in bread, carbs in bread. So the cutting of sugar, that's what glycolysis or glycolysis is. So I have this just cartoon 6-carbon sugar, and at the bottom, you end up with 2-3 carbon sugars. They're called pyruvate. All right, a couple things that happen. We have NAD plus reduced. And so again, if you were not in my last stream, we talked about the acronym oil rig. So oxidation is loss of electrons, and then reduction is gain of electrons. Okay, so NAD plus, it's plus because it's missing an electron, right? So we think about electrons are negative, right? So we need, once you add one electron, it would just become NAD, and then we add another electron, it's H. Okay, H meaning just a hydrogen atom, and remember hydrogen atoms have 1 proton, 1 electron, so it's going to carry an electron with it. And so it kind of looks like a brown suitcase almost with 2 blue circles. Those are the 2 electrons that are now in the carriers. These carriers, I would say, are the most important thing to kind of pay attention to in cellular respiration, because if we're following the electrons, then we know where the energy is going. So glucose is broken up. It takes a bunch of different steps. A little bit of ATP is made. And we have NADH and 2 pyruvate molecules. So that's kind of the gist of glycolysis. Another thing that would be important to know in terms of its linkage to evolution is that it's the most evolutionarily ancient pathway. So we could look back at organisms that have been around a really, really long time. So things like photosynthetic bacteria, or just lots of different single-celled organisms that have been able to do glycolysis for a really long time. Another reason we say it's kind of evolutionarily ancient is that all organisms can do this. And so generally speaking, if all organisms can do this one thing, like how all organisms have DNA or RNA, we'd say it's pretty old, right? Like it's been around a long time. This process is not something that has derived recently. Okay. Another big thing about glycolysis is that it can happen without mitochondria and without oxygen. All right, so big things to know. Glycolysis, and actually I'm going to delete this pyruvate oxidation. Just we're looking at glycolysis right now. Oh, wait, just kidding. The picture is of pyruvate oxidation, sorry. So what goes into glycolysis? Glucose, right? What comes out of it? A little bit of ATP, the electron carriers, which are your NADH. It's an electron carrier. It carries electrons. And then we also have pyruvate attached to an acetyl-CoA molecule. So it's a little carbon molecule with an enzyme attached to it. And that enzyme will allow the process of Krebs cycle to happen. Okay, so this ending thing, acetyl-CoA, it's an enzyme that's attached to the molecule. And then that enzyme will allow the changed pyruvate to enter the Krebs cycle. Okay, so those are just big picture glycolysis stuff. When we do not have oxygen, because again, I told you you don't need oxygen to do glycolysis, but if you're an organism that normally needs oxygen, what your body would do, what an organism would do without oxygen, because it's going to need a lot of ATP. It could do one of two methods of fermentation. So it can either do alcohol fermentation or lactic acid fermentation. So you can kind of see here from top to bottom, similar to my last diagram, we have glucose being made into pyruvate, right? But then the reason it can't just stop there is that those electron carriers and ATP need to be replenished. Oh, yes. Yeah. Active transport uses ATP. Yes, absolutely. Good. So yeah, ATP is super important, right? So if you're an organism that normally uses, does a full cellular respiration, you need a lot of ATP, but for some reason there's no oxygen, it can go through lactic acid or alcohol fermentation. So for example, this happens in our muscles a lot, where the amount of ATP we are using is greater than how much we're making. And so your muscle cells that have basically run out of oxygen will continue to make ATP using lactic acid fermentation. And lactate is what is your product here. But again, the big purpose of this, in terms of what you should know, you might be looking at this like, what the heck am I supposed to know from this? Two things. One, these are the things that happen when there's no oxygen. Okay. Number two, the reason this is happening is that, and why we can't just end at pyruvate, is that those electron carriers need to be recycled. It's kind of similar to how an enzyme, once it's been used, it can be reused again. Well, that NADH that was reduced, it needs to be oxidized so it can be recycled. So electrons are given to it, but because it has nowhere else to go, it's not going to go to the Krebs cycle and then the electron transport chain. We need that electron carrier to be recycled so that more glucose can be broken up. Okay, so those things need to be recycled. And so that's why that extra step of fermentation happens, where ethanol is made or lactate is made. So those are your two big things you should know. All right, I have another concept check for you. So if oxygen is not present in sufficient quantity, pyruvate molecules are converted into, what are the two things that pyruvate could be made into so that we can recycle our NAD. One happens in the muscles. One happens in things like yeast. They start, the process is fermentation. So just type it in. What's one thing? Okay, awesome. Thank you, Soleil. Good. So Soleil's right. So lactic acid and alcohol would be made, depending on what organism you are, right? Lactic acid would happen in our muscle cells, where alcohol fermentation is happening in organisms like yeast. So, if someone is brewing beer, for example, they have yeast in there in the solution, and then they trap it so there's no oxygen, and so a bunch of alcohol fermentation happens, and then that's where the actual alcohol content comes into play for alcoholic drinks. Okay. All right, step number 2. So that was glycolysis. Step 2 is our citric acid cycle or Krebs cycle. So I have the title as Krebs cycle, but then the diagram has citric acid cycle at the bottom. So just be aware, again, it's the exact same thing. It's just named 2 different things, which is kind of a pain sometimes in biology, I know, but just know them both. I would recommend just knowing them both. Okay, so this cycle right here, we're inside the mitochondrial matrix. So we came from the cytoplasm. We've now crossed the mitochondrial membrane, and we're inside the mitochondrial membrane, that inside portion of the mitochondria. And this diagram is nuts. If you saw it and you were like, what in the world? This is another example of something that many years ago you would have had to know step by step what's happening, where the carbon's going, where the bond's going. You don't have to do that anymore because it's just regurgitation. It's not learning. So big picture stuff that you do need to know is, first of all, what is going in and what is going out. So knowing that your acetyl-CoA kind of goes in here, but then another big focus, oh good, I did type it. The big purpose here of this entire cycle is to load up electron carriers. So you can see here in red, we have NADH, 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 right? So that's just 4 molecules right there for just one pyruvate changed molecule, right? So we know 2 pyruvate molecules are going to be going in. So we're going to have 8 NADHs filled. And then there's also another carrier called FAD, and you can see it here, FADH2. Oh, so I'm sorry, so 6 NAD and 2 FADH2s. Depending on your teacher, they may have you memorize exactly how many NADH and how many FADH2 carriers are being loaded up. But hopefully not, because that's not super important. It's just knowing, okay, we have this citric acid cycle. We make a little bit of ATP, tiny bit, but the biggest thing here is we are loading up electron carriers, and we get electrons by reconfiguring all of these molecules. We're going from one molecule to the next, to the next, to the next, and every reconfiguration, we're able to pull electrons off of a molecule because we were able to take away carbon, right? So CO2 would be made so that we don't need to worry about where the electrons are coming from. Okay, so another big picture thing, what goes in? We have pyruvate going in, it's the Krebs cycle. Technically, it's changed pyruvate. I guess I should have written that it's just 2-acetyl-CoA, but it's fine. What comes out? A little bit of ATP, electron carriers. That's our biggest thing, electron carriers. So again, this is actually a diagram that the AP College Board drew to simplify Krebs cycle. So if you're worried about that last diagram I showed you, this diagram is one that the AP College Board drew. And so hopefully you're able to see like, okay, that's actually not a lot of things. It's not too scary. So CO2 is released, electron carriers are loaded up, and a little bit of ATP is made. So I would encourage you, when you are studying this stuff, make a list of where everything's happening, what's going in and what's coming out. And it'll help you tell the story of cellular respiration. So don't just memorize it, but be able to tell the story of, okay, well, oxygen comes in, but have we even used oxygen yet? No. Okay, so just keep thinking of things like that. Okay, concept check number 3. Just kind of alluded to this, but that's all right. The NADH and FADH2 that are formed in the Krebs cycle primarily function to A, remove waste product, CO2, from the system. B, what is this? Oh, there we go. B, facilitate the production of water by combining hydrogen ions with oxygen. catalyze the production of ATP from ADP, provide energy to synthesize acetyl-CoA, or transfer electrons to the electron transport system. And again, here is important to not just memorize NADH and FADH2, but to recognize, okay, this is a molecule that has taken electrons, right? 2 electrons each. And so those electrons are energy for the ETC. So yes, good. E is your answer. It's to transfer electrons to the electron transport system. So Krebs cycle, we are loading up electron carriers. We are taking electrons. We are oxidizing all sorts of things. Remember, oxidizing means losing electrons. So we are taking those electrons and then we're moving them over to the electron transport chain. Alrighty, so here's a picture of electron transport chain. At the very end of the chain is our same enzyme from photosynthesis, ATP synthase. So again, another enzyme that's important. ATP synthase, it's synthesizing ATP, making ATP for us. Before we do that, we have to power this ATP synthase. It's almost like like a water turbine, right? Where water comes in and it spins it around for energy, or you could think of a wind turbine even. So ATP synthase physically spins within the membrane of the mitochondria, the inner membrane. So you can see over to the right over here, I'm kind of going at the end of ETC here, but ATP synthase works by phosphorylating. That's why it's called oxidative phosphorylation. It's phosphorylating ADP, D meaning di. There's only 2 phosphates. You can see it here. There's 2 little circles. And it's grabbing another phosphate and putting it onto the molecule. Okay. The reason it can do that is because there's a ton of hydrogen ions in the inner membrane space. And so once there's a nice concentration gradient of hydrogen ions, they'll flow through naturally, right? Just by diffusion, high to low. So once there's enough of a concentration gradient, those hydrogens are going to fly through. And that is kind of like the wind turbine idea. It's like wind going through this spinny thing. So ATP synthase can only work and do its job if there's a high concentration of hydrogens. So how does it get a high concentration of hydrogens? Okay, so that makes us kind of go back to the beginning of electron transport chain. Chemiosmosis is, if you think about osmosis, right, it's the movement of water, but here we're talking about the movement of just an ion. So in order to make that concentration gradient, we need to pump hydrogen ions from the matrix outside into the inner membrane space. So we're still in the mitochondria. We're going from the very, very inside of the mitochondria to that inner membrane area. Okay, and the way we do that is using those electron carriers. So each carrier shows up and basically gets oxidized, right? So we go from being NADH to NAD+. It's lost its electrons. And so now the electrons can fall through this electron transport chain, and it's all about electrons dropping energy levels. And when it does that, that energy can provide the push for hydrogen ions to be pushed up into the inner membrane space. Okay, I would say the electron transport chain of cell resp is probably one of the most misunderstood or most confused parts of cellular respiration for students. So that's kind of why I like to work backwards in terms of, okay, I know at the end of this, a ton of ATP is made. Well, how does that happen? So I kind of start on the right and think about, well, ATP synthase has to move. Well, how does it move? It needs a concentration gradient. Well, how does it get the concentration gradient? It gets it from those original electron carriers that dropped off their electrons, okay? The last thing about the electron transport chain and the phosphorylation is that there needs to be some sort of pull. Like you might ask, well, why, the electron carriers are dropping these electrons off, but why, what makes them fall down the energy levels? Like why don't they just hang out? If you look at this diagram here, we have oxygen. Oxygen is waiting at the very end of the electron transport chain. And if we remember back from the very first unit of AP Bio, oxygen is extremely electronegative. So it loves electrons. So it's actually pulling those electrons to it because it wants them so bad, right? So and electrons have this pull from oxygen. And oxygen is, we call it the final electron acceptor. And when that happens, when it's finally accepting an electron, that's where water's made. Okay. So without that pull from oxygen, this entire chain doesn't work. Okay. So that's why, like, even if for a few minutes, let's say somebody is without oxygen, if a human is without oxygen for a couple minutes, Think about what happens to their brain cells, right? If those cells can no longer do cellular respiration and make ATP to do the cell's work, without that oxygen being there as the final electron acceptor, that cell is going to perish. That cell will die. And that's why oxygen is so important to us, right? We have to breathe. Not breathing is a really big deal, and can be very detrimental, especially to our brain cells. Okay, so I know that was a lot. Oh, sorry. So again, like I've done on the other ones, we think about what goes in, electron carriers, and oxygen. So that's the big things of our electron transport chain. And then what comes out, big amounts of ATP and water. All right, I have one more picture of the electron transport chain that shows the whole thing. So you can kind of see NADH on the left and FADH being reduced, oxidized. So they're losing electrons. And so those protons are going to go, or those hydrogen ions are going to go through the inner mitochondrial membrane and hang out in that inner membrane space, which is the space between the outside of the mitochondria and that inner membrane wiggly thing. So again, that's why drawing diagrams helps, because sometimes when you're zoomed in on this, you really understand what's going on, but then you're like, well, where am I in this mitochondria? So if you need to, draw a diagram. Anyway, I also like this diagram because it kind of shows how, two hydrogen ions and two electrons can kind of come together with your oxygen to make H2O. And then here it's also showing the phosphorylation of ADP to make ATP. Okay, so I just like that diagram. I think it's good. Okay, that's it for like my lecturing and talking. I have a couple practice questions and then for response. And then if there's any questions from you, we can do that too. So number 4, this is the thing I didn't talk about explicitly, but that's what AP Bio is all about, is that sometimes the questions you see were never explicitly talked about in class, right? But you should be able to think things through and come up with an answer. So this question says, the inner folded membrane of the mitochondria serves primarily to A, separate the many reactions that occur in the organelle, B, provide increased surface area for the molecules and reactions of the electron transport system, C, provide a space for the reaction that unites acetyl with coenzyme A, B, compartmentalize the diffusing waste products from the cellular respiration reactions, E, produced enzymes that facilitate the splitting of water for oxygen release. Yes, good. It's B. Good job. Absolutely. So I don't think I talked about surface area. I think I said something about surface area in photosynthesis. But absolutely, those inner folds are there for a reason. They're just like the folds of your intestines, right? Your small and large intestines have all these crazy folds. And why do they have those folds? To increase surface area so that nutrients can be absorbed. So exactly like that, it's the same structure function. The structure is this folded membrane. The function is to increase surface area. This is a theme seen throughout biology. Okay, if you see something that has foldy stuff going on, it's to increase surface area so more stuff can happen. Okay, so very good job. All right, another multiple choice. The final or terminal electron acceptor in mitochondrial respiration is... So what is the final electron acceptor? We have A, hydrogen, B, NADH, C, pyruvate, D, oxygen, or E, carbon dioxide. So it's basically the very last thing in your electron transport chain. It provides that pull for all the electrons. It's the final electron acceptor. Without it, there would be no electron transport chain because the electrons would just never be oxidized in the first place, or the carriers would never be oxidized in the first place. It is oxygen. Oxygen is our final electron acceptor. All right. I think I do have one more multiple choice. Yes. Okay. This is similar to a test question that I used to give to my students. I don't use my test questions for 5-level just because I don't want my students to see them all, but it's a similar question. So which of the following represents a plausible pathway of electrons through the process of cellular respiration? So if we were to just say, let's say we were to like tag one electron, with some luminescence, right? If we tagged one electron, which order of things would it actually hit? So this one's kind of tough because this is not exclusively showing all the steps, but which one makes most sense. So I'll give you a second to just read through that. A is showing that electrons start with glucose, then pyruvate, then acetyl-CoA, then they could possibly go to FADH2, and then they could possibly end with oxygen. So think about A, B, C, or D, and all those different options. Which one makes most sense in terms of where electrons go? Okay, so I'm going to go over from bottom. I'm going to go over why certain ones are wrong. Okay. So for example, D is incorrect because it's showing electrons going into carbon dioxide. But carbon dioxide is produced in the citric acid cycle. So if that was the end in terms of like where electrons go, or actually even it's just out of order, carbon dioxide, that's released way before the electron transport chain. So D cannot be right. C cannot be right because acetyl-CoA also is something that happens before the electron transport chain. So C cannot be right. B, glucose to NADH to ETC. Here we go again. We're out of order because acetyl-CoA is happening before, it should happen before the electron transport chain. So the only one that is right is A. So A is the only one that's right. The reason why a lot of you probably felt unsure about A is because it didn't have the name electron transport chain in there. But again, electron transport chain is just one whole section of stuff that's happening. Really, if we're following an electron, it's going from an electron carrier, it goes through the electron transport chain, sure, but if it goes from carrier to oxygen, that's where it ends. So it's okay if it's not in there. Okay. Free response question. I know we're kind of going long tonight, but this is a really big topic. So we'll just do this one free response. All right. So organisms who require aerobic cellular respiration to survive depend on the presence of oxygen. Step by step, explain parts of cellular respiration that will fail to happen when oxygen is not available. So I'll give you a hint. And you don't have to type anything up in the chat box. You can just kind of jot it down on a piece of paper. So jot down from end to beginning, what won't happen if oxygen is not available. True. So some organisms could just undergo fermentation, absolutely. But let's say this is a human cell. So we answer this as if I'm talking about like a human brain cell. Okay, just for the interest of time, I'm going to jot down some ideas. So without oxygen, I'm just going to type it. Yeah, absolutely. So number one, only glycolysis can occur, right? But also explain why. So I kind of like to go from the end to the beginning. So if there's no final electron acceptor, the electron transport chain comes to a halt. Okay, and so kind of explain what does that mean if it's coming to a halt. So that means no water is going to be produced. Another thing is that NADH and FADH2 are not oxidized, right? So their electrons aren't going through the ETC. And so if those aren't being oxidized, the recycling of the electron carriers doesn't happen. And so because of that, the Krebs cycle would also come to a stop. Right? So without empty NAD and FADH, Oxidizing of your carbon molecules can't happen. All right. So again, this is just a FRQ that I wrote. It's not one from an AP exam. But the next one, I'm not going to go over these two right now. I'm going to hold these off for that day that I talked about, where I'm just going to do an FRQ. So this is a free response question from the 2015 AP exam. So those are online for free. AP College Board publishes them. So you could look it up. It's the 2015 AP exam. I don't remember, I can't remember which question it is. But there's 4 parts to it, A, B, C, D. Be a really good review. So I encourage you to check that out before the stream, and then keep your eye out for the stream, and you know, come prepared, obviously, because I'm not going to spend the entire time. Can we hit 1 hour? Yeah, we can do 1 hour. So if you guys have any questions, it's 2 minutes. If you have questions, shoot them out. Those of you who have never come to my streams, I just had a baby 1 month ago, so I try to keep my streams to about 50 minutes, just to kind of maintain a nice work-life balance. But this was a really big topic, so no questions? Oh, sorry, Joseph, we're not going to hit the hour. That's okay. All right, if there are no questions, because I don't see any in the question box, you guys are good to go. Okay, so again, check out, go to the, Google 2015 AP Bio FRQ. What's in NADH? So NAD, it's actually just a molecule called NAD. You do not need to know the chemical molecular configuration of it at all, but basically it's just a molecule that has the capacity to hold 2 extra electrons. So it has an affinity for electrons. So Lei just asked a question, what's in NADH? So if we're calling it NADH, that means it's the reduced version of NAD+. Okay, so it's been filled with electrons. So I would call it, it's a filled electron carrier, NADH. Okay, all right. Thanks for coming, everybody. Again, keep your eye out for when we're going to do another, like a cell resp, photosynthesis, ask me anything stream. So you can come with this free response question kind of thought out, and I'll go over it. And we'll also just kind of ask any questions that you might have. We hit 1 hour. Yes, we did. Okay. All right, everybody. Thank you. Have a good night. We'll see you next time. See ya.